right, welcome to the Explainer. If you're studying for your medical exams, you know just how critical high-yield facts are. So today, we are going to break down everything you absolutely need to know about COVID-19. We're going to get it done in the next 5 minutes. Let's go.
Okay, let's just dive right in. First up, our subject, SARS-CoV-2, the virus behind the whole COVID-19 pandemic. Now, take a look at this electron micrograph. You see those little projections on its surface? They create this crown-like appearance, which is exactly where the name coronavirus comes from. You know, like a solar corona. Pretty cool, huh? So here's our game plan for today. First, we're going to look at the enemy's blueprint, its morphology. Then we'll get into its attack strategy, the pathogenesis. After that, we'll cover how we detect it in the lab. And finally, we'll go through our complete defensive arsenal, vaccines, treatments, and prophylaxis. Let's get started. All right, Section 1, the blueprint. Look, understanding the virus's morphology is absolutely fundamental. If you can get this right, I promise the rest is going to make perfect sense for your exams.
This diagram right here, this is key for your exams. So let's break it down. First, notice that outer layer? That means it's an enveloped virus. Now inside, its genetic material is positive-sense, single-stranded RNA, all protected by a helical capsid. You got to remember those three features. The whole thing, by the way, is about 120 nanometers.
Now let's focus on these 4 key structural proteins. Think of them as the virus's essential tools. The spike, or S protein, is literally the key that unlocks our cells. The membrane, or M protein, is the most abundant one, and it gives the virus its overall structure. And then you've got the envelope and nucleocapsid proteins, which are not only vital for the virus's life cycle, but are also key targets for our diagnostic tests.
Okay, Section 2, the attack strategy. So, how does this structure actually translate into disease? Let's break down the pathogenesis.
The pathogenesis really is this devastating 5-step cascade. It all starts with Step 1. The spike protein binds to ACE2 receptors on our lung cells. Step 2. This damages those cells, which reduces surfactant and causes the alveoli to literally collapse. Step 3. The body freaks out and responds with a massive, uncontrolled inflammatory reaction. You've heard of it, the cytokine storm. And that storm, in Step 4, leads to severe lung damage, which all culminates in Step 5, acute respiratory distress syndrome, or ARDS, a state of really profound respiratory failure. And this diagram, this is a perfect illustration of that cytokine storm. I mean, look, on the left, you've got a nice healthy alveolus, but on the right, you see the result of the storm, this flood of cytokines causing vascular leakage, massive inflammation, and even blood clots. Trust me, being able to explain this visual is a classic exam question.
So, Section 3, detection. We know how it attacks. Now, how do we actually find it? Let's cover the high-yield points of lab diagnosis.
You know, mnemonics are absolute gold for exams. To remember the key gene targets for RT-PCR screening, just think SEM. That stands for spike, envelope, and membrane proteins. These are the main genes we're looking for. You might also see the mnemonic SAME used. The A is really just a placeholder to make the word easier to remember.
Now this is what a positive RT-PCR result actually looks like. See these curves going up? They represent the amplification of specific viral genes, like the envelope gene and the RDRP gene. That's the RNA-dependent RNA polymerase. When these curves shoot up and cross that threshold line before 40 cycles, that's a positive result. Simple as that.
This table is extremely high yield. You absolutely have to know the difference here. So NAAT, or RT-PCR, is the gold standard for diagnosing an active infection. Antigen tests, they're for rapid screening, usually in the first week of symptoms. And antibody tests look for past exposure. They tell you if a person was infected, not if they are currently infected.
And this timeline is so crucial for understanding the diagnostic windows. Just follow the blue line. See how viral RNA is detectable first, right at the beginning of symptoms? Next, look at the green line. That's your initial IgM antibody response. And finally, the red line shows the long-lasting IgG response, which gives you that longer-term immunity. Knowing this sequence, RNA, then IgM, then IgG, is absolutely essential. All right, Section 4, the shield. Let's talk about our primary line of defense, the vaccines that were developed to fight this thing. For your exams, you have got to know these 3 vaccine platforms. First up, inactivated vaccines, which just use a killed version of the virus. Second, you've got viral vector vaccines, which use a totally harmless virus as a little delivery truck for the spike protein code.
And third, RNA vaccines, which use mRNA to teach our own cells how to make the spike protein. Know the mechanism and an example for each one.
Now, in the Indian context, the two main vaccines to really remember are Covaxin and Covishield. Covaxin is an indigenous inactivated virus vaccine. Covishield is a viral vector vaccine. Make sure you know which one is which. That's a classic question.
Okay, Section 5, the counteroffensive. What if someone is already sick? Let's take a look at our treatment strategies for managing the disease.
Well, treatment is really multifaceted. It all starts with the foundation, supportive care, things like oxygen therapy. But for more severe cases, we bring out the targeted therapies. One is remdesivir, an antiviral that directly stops the virus from replicating. Another is tocilizumab, which is a monoclonal antibody that targets the IL-6 receptor to calm down that cytokine storm. So remember, one attacks the virus, the other attacks the inflammation.
And finally, Section 6, the defense plan. This is arguably the most important strategy of all, preventing infection in the first place through prophylaxis and control measures.
These infection prevention and control measures, or IPC, they are the absolute bedrock of our defense. You have to remember the essentials: good hand hygiene, social distancing of at least 1 meter, and proper mask use. These are, by far, the simplest and most effective tools we have to break the chain of transmission.
And for all you future healthcare workers, personal protective equipment, or PPE, is non-negotiable. As this slide shows, the standard PPE includes a gown, gloves, eye protection, like a face shield, and a medical mask. But, and this is important, remember that for any aerosol-generating procedures, you must upgrade that to an N95 respirator.
And that is your high-yield review of COVID-19. We've covered the virus's blueprint, its attack strategy, and our full defensive playbook. So the final question to leave you with is this: how do we take all of these incredibly hard-won lessons to better prepare for the next pandemic?